Hi everyone. I might just wait for one more minute to see if more people join, and then we will start the webinar. So I think while we wait for people to join, I'm just going to talk a little bit about sort of myself and my experience with this microscope while we wait. So this here is a transmission electron microscope. So we're here at the Centre for Bioimaging at Oxford Brookes University in Oxford. And I am the manager of the Centre for Bioimaging, but also I'm the lead electron microscopist at the Centre for Bioimaging. So I do a lot of the electron microscopy here, and I sort of supervise people that do electron microscopy. This microscope was actually a microscope like this one, a transmission electron microscope, was actually the first one I ever used when I was an undergraduate many years ago. I was about 18 years old. I used this microscope for the first time, and it was absolutely awesome. This is an incredible microscope. It was a real treat. So when I was a secondary school student, I really fell in love with cell biology. I loved, seeing all these pictures of the cell inside. And this is the microscope where you can see all of the components of the cell inside and all of the tiny little details of these things. This is a very, very powerful microscope, and I was just completely in awe with it, that you could see all these details. So today we are going to see what it's like to prepare samples to go onto a microscope like this, and then to see samples to look inside cells and tissues in a microscope like this. Okay, so how many minutes have we got? So we've got for a couple of minutes. So I think we can start the actual webinar now. So this is the microscope here, but the first thing I would like to do with you is actually show you how we prepare samples to go on the microscope. So the scale of what we're looking at here is very, very, very small, much, much smaller than the scale that we can see in the naked eye. So to show you how to prepare samples, how we prepare samples to go on this microscope, I'll actually have to use another microscope, a smaller one, a light microscope, that will show you the very, very small samples and the details of the sample prep. So my colleague Ed here will move the camera to a different position where we have an area with lots of bits to show you how to prepare samples. And then we're going to go back here, and so we'll be repositioning the camera again to show you once the sample goes inside the microscope and looking at the sample down the microscope, okay? One thing I'd like to say is that this is a webinar, but please feel free to ask questions. I believe you have to raise your hand, and then we will let you in. We will unmute you, and then you can ask me questions, okay? So the first thing we need to do, so imagine, we're putting samples, very, very small samples into the microscope. So the first thing we need to do is to actually get the sample in a very, very small bit. So it could be, let's say, a very small piece of a leaf, for example. We deal with biological samples here, so it's all like tissues of animals or plants. So we could have, for example, a very small piece of an animal tissue. So it could be a very small piece of a biopsy. So something that the doctor's gone and had to take a little piece of a patient, and then we cut these pieces in very small bits. And then we do the first step, which is what we call fixation. Yeah. So we put that little piece of tissue in a liquid that will fix it in a state that's very close to the way the tissue was when it was alive. Yeah. So fixation will first kill the tissue, obviously, but it will also fix it in the right position so that we can then image it. Yeah. To give you an idea of size, so this here in my hand, can you see this is a tube? This is a very small tube, and in here there are some very small pieces of tissue. You won't see them here very well. I'm going to put them under the microscope. So we have another microscope here. That's a little light microscope, and it has a screen attached to it. So let's put the screen. Yeah, there you go. So now I'm going to put this tube under the microscope, and you will see the pieces of tissue that I'm talking about. They are quite small. So these here are the pieces of tissue, okay? So they're very, very small, and they need to be very small for this type of technique. Can you see that the tissue is sort of a little bit pale? Yeah. So normally animal tissue does look a little bit pale when it's fixed, unless it has some, some natural pigmentation, which this one has a little bit as well. Plant tissue might look a bit green, obviously, because it has natural pigmentation of chlorophyll. And then the second step we have to do after we fix the tissue is a step of staining. Yeah, it's called staining. During staining, the tissue will become black. Yeah. So in this block here, this is a little resin block, and at the tip of the resin block, we have a piece of tissue that has been stained. The piece of tissue has been stained with lots and lots of heavy metals, and hopefully you'll be able to see better on the microscope here the piece of tissue stained. Let me see if this light helps as well. So here at the tip, this dark spot, that's that piece of tissue that became dark because we have impregnated lots and lots of heavy metals on the piece of tissue, so it's dark. The heavy metals are very important to give contrast to the sample. So in electron microscopy, we don't talk about things becoming colorful. Yeah, so in light microscopy, we tend to have color. In electron microscopy, we have contrast. So some areas are darker than others, and that's what makes the image. And this piece of tissue here, it's dark now, so it will give us good contrast when we image. Yeah. So we've been through the steps of fixation and staining, and now I need to tell you a little bit about another step called embedding. Okay. So once we have the dark piece of tissue, can you see that this piece of tissue is actually inside a little block? Yeah. So this block, it's called a block of resin. I'll put it under here so you can see. Yeah. So this is a block of resin. What we do in electron microscopy, in transmission electron microscopy, is we have to take sections of the sample. Yeah. And we can't take sections of a piece of tissue that's all nice, mushy and squashy. It's very difficult to take sections of it. So we have to embed the tissue in a nice hard matrix. And this hard matrix is this block of resin. So we embed the tissue in resin. And then the final part of it is what we call sectioning. So once we've been through all these steps of fixing, staining, embedding, we then have to section the tissue into slices. So what happens in transmission electron microscopy? In transmission electron microscopy, the beam of electrons will go through a slice of the sample. And just to give you an example of sort of how this microscopy would work, if it was something, in the naked eye, I've got here a slice of an orange. Yeah, so this is a slice of an orange. In this slice of the orange, we can see the inside of the orange. So we can't see the entire orange, but if we shine a light on this slice of orange, we can see the inside of it. Okay, so here the microscope is showing the inside of the orange. So that's the same that we're going to do with the tissue for the cells. Okay, so we have a piece of tissue with lots and lots of cells, and we are going to look at a section of it. The electron beam is going to go through that section, and we'll be able to see the inside. But obviously, the piece of tissue that we have is much, much smaller than this. So it's our nice little block here. And it is actually just the very tip of this little block. How do we cut a section, a very, very thin section of the tip of this little block? And when I talk about thin sections, I'm talking about really, really thin sections. So this is in the nanometer scale. So the sections we look at down the electron microscope, they're about 70 nanometers in thickness, 70 to 90 nanometers in thickness regularly. To cut a section of this very tip here, a section that's this thin, we need a special piece of equipment called a diamond knife. And that's a diamond knife here. And I'm holding it very carefully because the diamond knife is called a diamond knife for a reason, because it has a diamond. At the very tip here, I'm going to show you the diamond. It has a diamond, a very sharp diamond. Okay, so I'll put the diamond knife under the microscope now. And hopefully we'll be able to see it. If you keep looking down here, this is the diamond of the knife. I'm going to put it in focus. There we go. Focus. And then I will move the knife around so that the diamond, hopefully some light will shine on the diamond. Is this on? Is this on? There we go. Sorry. Yeah, can you see here? The tip of the knife has a diamond here. Yeah. And this diamond has a very, very sharp edge. From the sharp edge of this diamond, we can cut very thin sections. Yeah. We actually have a little video, don't we, Ed?
Yes.
Of the diamond knife cutting sections of a block of resin. So we will cut out to the video now. So this video is showing the diamond knife cutting sections of a block of resin. And we can see this diamond knife through a microscope because it's very small and the sections are also very small. Okay. So once we have our sections that have been cut using a diamond knife, then we collect the sections on a small metal grid, very small metal grid. And I've got the grid here with me. I'm going to show it to you down the microscope. Next to this grid is a coin, and you can see how small the grid is, yeah, compared with the coin. Let me show you the grid down the microscope. Let's turn it around. Okay. Find the grid. There's the grid there. Let me put it in focus. Middle. I hope you can see that in this grid, right in the middle here, there are some sections. So these pale areas here are these sections that are so thin that they just look like a thin kind of veil on top of the grid, yeah, of the metal grid. And this grid is about 3 millimeters wide. So we are making it look much bigger up on the microscope, but it's tiny, tiny. Okay. Once we have the sections, once we have the section on a grid, once we have the section on a grid, we put the section, the grid on a holder. And this holder will allow us to put the section on the microscope, okay? Yeah, so this is the tip of the holder that will go into the microscope. So this is the sample holder that we will use to insert the sample on the microscope. At the very tip here, there is a little grid, yeah? So let's move to the microscope. Just to say a few words about the different parts of this microscope and how it works. So at the top of the microscope right here, you can see this big cable going onto it, big electric cable. At this part of the microscope here, there's an electron gun. So this is a part of the microscope that generates a very, very strong electron beam, so a very strong beam of electrons. The beam of electrons travels down the microscope inside this reinforced bit here called a column. Yeah? The column is all reinforced, and you don't see the beam traveling in the microscope. The beam is all inside the column. And the column is reinforced because it's all under vacuum. The vacuum is really important to make sure that the beam travels nice and straight, because if you have lots of atoms inside the column of the microscope, the electrons will hit all these atoms and will get, distracted, go the wrong way. We want a beam that's nice and straight and strong, traveling down the column of the microscope. So the beam generated up there, it's going to travel all the way here, and down here, right here in the middle, we are going to insert our sample. The beam will go through the sample. So this is called transmission electron microscopy, because the beam is transmitted through the sample. Okay? So the beam will go through that section of sample here, and then it will project an image of the sample on a screen here. In practice, we no longer look at samples here very often. We then go to the camera, we project the images to the camera, and you will see that. But this is the screen of the microscope, where we can actually see the sample down here on this screen. Okay. So now we're going to put a sample in here. We're going to introduce a sample into the microscope. So I mentioned to you that the microscope, the column is all under vacuum, which presents a little bit of a problem, because if we want to put a sample inside the column, if we just insert it straight in the column, it would break the vacuum of the column. Yeah, so the column needs to remain in vacuum while we insert the sample. To be able to do that, we have the holder that we use for inserting the sample. It's a special device. It's a little bit like an airlock device. If you've seen like science fiction films, people coming in and out of spacecrafts, yeah, through this airlock system. So they have a special room that, you know, builds up pressure. This is a bit similar here. So this has an airlock system that when we insert the sample will go to an intermediate chamber where vacuum will build, and then the sample will go inside the microscope. Yeah, so it's a special airlock system. How are we doing time-wise? Okay, so I will get the sample holder and then bring the sample holder here to insert the sample. Oh, excellent. I've got Ed brought the sample holder for me. Okay, so now we have the sample holder at the tip there. We have our sample, and the sample is going to go inside the microscope. Let's put the sample in. And now we are telling the microscope to pump this area because we are about to put a sample inside. And here on the computer that controls the microscope, so this microscope is attached to two computers. One computer controls the microscope itself, and the other computer controls the camera that we use to take pictures of our samples. So it's got two computers attached to it. And this computer here is telling me lots and lots of things about how the microscope is working. On this side here, it's telling me that the vacuum is good on several parts of the microscope, and it will tell me when the vacuum is good here, so that I can then insert the sample fully in the microscope. Very soon, hopefully, vacuum will be ready here, and then we can insert the sample. That's it. It's opening up the valves. Okay. The sample is in. And in about 30 seconds, the microscope will allow me to switch the electron beam on. So the electron beam is not on at the moment. The electron beam will come on when I tell the microscope to switch it on, which will be very, very soon as this goes green, and then it means it's all ready for us to image. The electron beam takes about 5 minutes to come on. And while it's coming on, I will show you some images that we've taken on this microscope. Now we can start the filament on, and then I will, that's the second position. I will sit down here, and while we wait for the beam to come on, what do you think may affect the image projection? So you mean what can affect the generation of an image? Is that what you mean? So what happens in this microscope, the way it works, so it works by contrast. So your sample will be impregnated with lots and lots of heavy metals. So we use in this microscope, we use heavy metals like uranium, osmium, lead. So if you know your periodic table, you'll see that these are all elements that have high Z numbers. So these are very, very heavy metals. And these heavy metals, they bind to all of the biological material. So they bind to membranes and things inside the cell. And when the electron beam hits them, the electrons don't go through where the heavy metals are. So this generates contrast. Yeah. Is that what your question is? What affects the image projection? And then So where the beam is going through is where you haven't got sample, and where the beam is blocked is where you have all the sample details. Yeah. Let me show you some images that we have taken on this one. So these are some images we've taken on this microscope. Let me just go back a bit. And you see some things that may be familiar to you guys. Parts of the cell that you might have learned about on your cell biology images. So this is Golgi here. It's gold. The Golgi is stained with a particular thing that goes inside the Golgi. So these are heavy metals that go inside the Golgi. So that only the Golgi appears dark. Yeah. So the electrons are blocked where the Golgi is. So these are all the Golgi stacks in here and vesicles coming out of the Golgi. So one thing I should point out to you, so you're not disappointed now, is that electron micrograph, so images taken in electron microscopes, they are all black and white. Because there's no color, it's all based on contrast, so images are all black and white. Of course, that doesn't stop us from being able to colorize the images after we take them. So sometimes you like putting a nice little blue one, or actually it's become quite an area now of like, you know, making electron micrographs colorful. So this is a piece of kidney tissue, and you can see various different parts of kidney cells in there. And another one as well. This here is a nucleus of a cell. So this here is a parasite cell. And it's got all sorts of interesting features. So it has all these microtubules here. Do you know have you heard of the cytoskeleton? So it's the skeleton that's inside our cells. And microtubules are an important part of the cytoskeleton. And here are the microtubules down here. There's more microtubules in here. Here is again more microtubule structure. So because it's a tube, if you cut the tube in cross section, that's what you see. You see a circle. Yeah. This here is actually a lymphocyte, so it's a cell of our blood, and you can see parts of the nucleus of this cell here and lots of mitochondria. This here are microvilli. We're going to see that in the sample that we'll look at now. So if you cut microvilli in cross section, so if they are obviously long and thin tubes, and if you cut them in cross section, that's what they look like. They just look like circles. And if you cut them along their axis, they look like this. Yeah, so along their length, they look like this. And I'll show you some of these structures down the microscope. So I don't want to spoil now. The microscope, the beam is nearly ready. So I'll stop sharing the screen, and then we'll start sharing the actual microscope capture screen. Capture screen. I hope you can all see this screen. So the capture screen of the microscope. I am going to have to turn the light off very, very briefly, but you will still see the capture screen. Okay. I first need to find the sample here, and then something. Lights back on. So now we are looking at a little piece of insect gut. So some researchers here at Brooks, they study diseases that are transmitted by insects. Yeah. Diseases like malaria, for example. And this particular piece of tissue was taken out of a mosquito that was infected with malaria. And actually, we probably won't see parasites in this piece of tissue, but we will see all sorts of interesting parts of the cell. Yeah. So just looking at a lower magnification first, so at a lower zoom, let's say. all of these circles here, yeah, they are all nuclei of different cells. So what you're seeing here is various cells that are very, very close together, okay? And when we zoom in, we will see that there is a boundary between them. Okay? So this is the nucleus of a cell, and this here is the boundary of this cell. The boundary is called the plasma membrane. And in actual fact, if we look closely, you'll see that the boundary of one cell is right next to the boundary of the next cell. These are plasma membranes here of two cells that are very close together. Okay? So the cells are all close together, and we are seeing a slice of them. Let's have a look around, and what I might do now is I might ask some questions. I might point at some structures, and I'll give you maybe 10 seconds to see if somebody tells me what the structures are, okay? I'm going to start with an easy one. This one here. I found a timer here. Focus. So you can, like in any matter, I've got time. So I'm going to put a timer on. There you go, 10 seconds. So in 10 seconds, have we got somebody telling me?
Yeah. 3 people have already.
What did they say? Who
They
said what?
So we've got, lots of people. Ben, Harney, Patrick, Jack, and Neha, and Alex have all said mitochondria.
Mitochondria, very good. That's an easy one, isn't it? So mitochondria have 2 membranes. Let's see if we can see the membranes here. So it's got an outer membrane. Got an outer membrane down here. Down here is the outer membrane, and then it's got an inner membrane. And the inner membrane is sort of folded inwards, isn't it? It's folded inwards, forming what we call the cristae, mitochondrial cristae. And you can actually see the cristae right here. You might be able to see a better one on another. Let's look around. This sample has some really stunning mitochondria around. Okay. So if we look in the middle of the cell, in this bit here that we call the cytoplasm, there are lots and lots of dots. What are these dots? Let's put the timer on. Oh, sorry. What are the dots? Who can tell me what the dots are? Lots and lots of dots.
Yeah, we've got some answers.
Yeah, well, the answers, who said what?
Sides and ribosomes.
Oh, ribosomes, very good. Very good. Yeah, excellent. And here we've got loads of ribosomes lined up on top of a membrane. What is that? Does anybody know? Ribosomes around the membrane.
Ben's got an answer for you.
Yeah.
He says it's the rough endoplasmic reticulum.
Oh, very good. Wow, we've got some cell biologists in there.
We do.
We have so much aspiring cell biologists, you know. Okay, excellent. So can you see here that there's loads and loads of rough endoplasmic reticulum? Loads and loads.
It's a bit slow. The camera feed a bit, a little bit
Is
slower. Just a tiny bit. It's okay.
Okay. Yeah, I'll make sure I stop. I
Yeah.
stop every now and again. Okay, so we've got more mitochondria. Let's here. Look at this very interesting endoplasmic reticulum. So this cell has, in the rough endoplasmic reticulum of these cells, they have this nice arrangement of lots and lots of circles. And you see it every now and again, a nice section through this type of ER. The ER in the plasmic reticulum is not always arranged like that. Here is a glancing section. So if you imagine that the ER has the membrane is like a sheet, yeah. If we cut across this sheet, then we can see all the little ribosomes that are on this sheet. So this is as if we were cutting a glancing section, like on my hand here. and then you actually see everything that's on, on the skin of my hand. So this is some ER. Let's find some other structures. I think I'm going to move to a slightly different part of this tissue, because I want to see if I find some microbes. So this is me moving around. If you remember when we looked down the microscope, down the little light microscope, and I showed you the sections on a grid. So these dark lines here, they are the actual, they are the mesh of the grid. So the mesh is so small that by eye you can't see it, but down the electron microscope you can see the mesh. Okay. Let's find some more structures. This here is a nucleus. And Ellie, are you there? I have lost Ed. Somebody distracted Ed. That's all right. Yeah.
Yes, I'm here.
Oh, it's all right. I was going to ask you to help me, but Ed's back. So the nucleus, this is the nucleus down here. Who can tell me how many membranes there are around the nucleus? I think Ed, are you able to help? Or
Yeah.
shall I wait? So I'll put the 10 seconds up. How many membranes are there around the nucleus?
We've got one answer.
Yeah.
We've
How
got
many?
two answers. So Harmony and Alex both say there's two membranes.
Two membranes. So the
And
nucleus
Deborah.
has. So I'm going round the nucleus here, and you can see the nuclear envelope, what we call the nuclear envelope. The nuclear envelope has two membranes, one outer and one inner. The outer membrane is towards the cytoplasm, and the inner membrane is towards the lumen of the nucleus, the inside of the nucleus. Okay. If we walk around the nuclear envelope, we can see that every now and again you find a gap. So there's a gap here.
Yeah, there you go.
Yeah. So there's a gap here, and then let's find another gap. There was a really nice gap. A second ago. When it starts, it's here. That's a gap. So what are these gaps in the nuclear envelope? What are the gaps? Who knows? Count the time.
Yeah, we've got one answer.
One answer. What?
Let's give them the full 10 seconds.
Okay.
And time is
Okay.
up. Oh, okay, so Ben says that it's nuclear pores.
It's the nuclear pore. Wow, you guys are good.
Yeah, I know.
Yeah, they are the nuclear pores. Yeah, so the nuclear pores, they control what goes in and out of the nucleus. Yeah, goes from the nucleus to the cytoplasm and back again. And they are not a proper pore, they are a complex. You can't quite see the complex here. Can you see that there is a gap in the nuclear envelope? But in this gap, there is something. Yeah, this gap's not empty, because in this gap, there is a complex of lots and lots of proteins that regulate what comes in and out. Yeah, so some very, very small things can actually just come in and out without, being stopped. But bigger things, they have to be checked. Yeah, at the entrance or at the exit of the nucleus by the nuclear pore complex.
Lovely. I've got to run 2 minutes,
Okay.
and then I'll be back in.
So Ellie, can you take over with making sure that, seeing if there's questions?
I will do.
Okay. Excellent. And inside the nucleus, what have we got? Another question. What have we got inside the nucleus? Who knows? Okay, time's up. Have we got some answers, Ellie? Can you tell?
We haven't got any answers for this one.
You haven't got any answer. You don't know what's inside the nucleus.
Oh, we have one. No, they're all coming in now, and everyone is saying DNA.
DNA. What else inside the nucleus? Who knows?
We've got chromosomes.
Yeah, so chromosomes, they have DNA and they have a lot of protein as well. Yeah. So the chromosomes in this cell here, they are not in the state that we call condensed. So they're a bit loose. Yeah. But there are parts of the chromosomes that are a bit more compact scattered. And in these parts are these dark areas here. Yeah. Some of these dark areas, they can have other specialized structures, so they're not necessarily just DNA and protein. They can have RNA as well. But yeah, the chromosomes are here. The chromosomes are DNA with lots of protein wrapped around it as well. And let's have a look. So this is some cells from the guts. And what do we find in the guts? We find these things here. I've actually shown them to you a little bit earlier on. So let's see who knows what these are. What are they? Have we got answers coming?
We've got Luke says that it's tissue, and then Ben says it's microvilli.
Yes, microvilli. Very good. These are microvilli. Look, they are these little fingers that extend out of the cells in the guts, and they make the area of the cell, the area that the cell has to kind of incorporate nutrients. They make that area much bigger, and so the cell has a bigger area to incorporate nutrients, So the microvilli, they are an extension of the cell membrane, and inside this extension, we've got some sort of stringy things here. Can you see? Can you see? It looks like a bit like little strings. What are they? Anybody know what they are? Inside the microvilli, it's like some stringy things.
They don't come across super well on Zoom, but you can't just about see
Yeah,
them.
but they won't come across.
You can't really see them that well on the camera, right?
Yeah, they won't come across super
We've
well.
not had any answers.
No, you've not got any answers. So these.
Oh, Harmony says, is it microtubules?
Nearly.
Nearly.
They are micro, but not tubules.
Alex says microfilament.
Well, I knew somebody would know. Microfilaments. Very good. They are microfilaments. So microfilaments make up part of the cytoskeleton of cells. So cells have this internal skeleton of different sort of filaments and tubules. And microfilaments are an important component of this skeleton. And here we have bundles of microfilaments that organize the structure inside the microvilli. Yeah. So the microvilli cuts in longitudinal section here, and then down here, because this is a section through a load and load of microvilli, we're going to catch them in different positions. So down here we've got cross sections of them like this, and then up here we've got longitudinal sections of them along their lengths. Lots and lots of them. And they are all at the top of the cell. Now let's look inside this cell. Very closely at the cytoplasm. So I want you to oh, there you go. There's one. We have here a long and thin structure. Let's see. It's quite faint. It's this one here. Can you see? Long and thin. Long and thin. Yeah. What is this here? You find little bits of it everywhere in the cell. So as you move around the cell, you find sections of parts of these things. What are they? Does anybody know? Nobody knows.
Yet.
So who talked about microfilaments earlier?
So Alex said microfilament,
Yeah.
but Harmony before had another answer.
Yeah. What was her answer?
Her answer is microtubules.
Microtubules. So this here is a microtubule. All around the cell, there are microtubules. These microtubules, they come from near the nucleus, the periphery of the nucleus, and they extend towards the cell membrane. Yeah, the membrane of the cell. And we obviously can't see this network here, but what we can see is that because they are everywhere, every now and again, as you look at the cytoplasm, you will see little sections of microtubules. Look, that's a section of a microtubule here. That's another section of a microtubule.
Can I ask you a question?
Yeah, go on.
How can you tell the difference between a microtubule and a microfilament?
Oh, the microfilament is much, much thinner than a microtubule.
Thinner even than a
Yeah,
tubule?
yeah. The microtubule is about 25 nanometers thick. And if you measure here, you will see. You can also see the microtubules have a wall. So it might be difficult for you to see if you're not used to looking. But on either side here, there's a wall. Yeah, so the microtubule is a tubule. So in each side, you will see kind of a bit more electron dense, the wall of the microtubule. The microfilament is very, very thin, and what you usually see are bundles of microfilaments. You can actually see them separately. Oh, these are nice mitochondrial crystals. That's a mitochondrion. You see? Yeah, sorry about the time that it takes sometimes for the images to appear. We have some other structures. We have a lot of other structures in the cells. So in here, there are lots and lots of peroxosomes which are involved in detoxing, so getting rid of things that are toxic for the cell. Cells have lots of different vacuoles. So when cells eat, the food that they eat goes into lots of vacuoles in a sequence, yeah? And this is one of these vacuoles. It's very difficult to say which type it is, but it's certainly one of the vacuoles where food goes in. Or it may be that this is actually a vacuole where the cell is eating part of itself to then recycle material inside the cell, because material is also recycled inside the cell. Okay. What else can you see here? Oh, this is interesting. This is, what do you think this is? I think this is a glanted section of a very special structure. But it might not be terribly obvious to you what it is in this particular section. Let's see if anybody has a guess of what this is.
No guesses yet?
No guesses?
10
No.
seconds.
So this here is a section through part of the Golgi complex. Has anybody heard of the Golgi complex? What does the Golgi do? Do people know what the Golgi does? Let's see. Somebody knows. It's a very special structure. It's got lots of vesicles. Can you see? There's lots and lots of vesicles around here. Lots and lots of vesicles because it does something very special.
So we've had two answers. So
Yeah.
Harmony says that it processes and packages proteins.
Whoa, very good.
And Ben says it produces vesicles to package molecules.
Yeah, exactly.
So it does both of those, really.
Yeah, so lots of proteins that are produced in a different organelle that I'm going to ask you in a minute what it is. Things are produced in a different organelle, they go to the Golgi, and then in the Golgi, these proteins are modified, and then they are packaged into vesicles. And these vesicles can go to different parts of the cell, or even to the outside of the cell. Yeah, so they can be secreted. And in these cells, we know there is a lot of secretion going on because cells from the gut lining, they secrete lots of mucus. So we know that they secrete a lot of stuff.
So that's another question I had. So this is a gut cell.
Yeah.
Would you find everything that you're looking at here, if you looked at any other cell in the body, would it look the same?
Very good question. And the answer is no. They wouldn't look exactly the same, but every cell has to perform a certain number of basic functions. So you would have all these structures in any cell, but they would most likely look a bit different. You would be able to recognize them. So be able to see, oh, this is Golgi, this is ER, this is whatever, mitochondria, but they will look different. And they even, between the different cells of the tissue, they will also look different. So let me see. So these things that are produced, the things that are packaged in the Golgi, they are produced somewhere else. Where are they produced?
The 10-second timer.
10-second timer.
Quick fire.
Slight hint on the screen, although probably not the best hint yet. Better hint.
Not had any answers yet.
Not had any answers. So
So the question is where
are proteins produced in the cell? Where are proteins produced in the cell? So the proteins are packaged in the Golgi complex. And
where were they made? In the
where are
first
they
place?
made? Where are they made?
So Harmony has got an answer. Alex has an answer.
Oh yeah, go
Yeah,
on.
now we get the answer. So Harmony says ribosomes or the rough endoplasmic reticulum,
Yes.
and Alex and Lewis both say ribosomes.
Exactly.
Yeah, lots of people say
So,
ribosomes now.
so the rough endoplasmic reticulum, the one that has ribosomes in it, it produces proteins that go into, usually they go into the endoplasmic reticulum or on the membrane of the endoplasmic reticulum, and then they get sent to the Golgi. So this is lots of ribosomes here on ER, but also like somebody who said that they are produced in the cytoplasm by ribosomes. So ribosomes can also make protein in the cytoplasm, obviously. They don't need to be attached to the ER to make protein. So that's what we call polyribosomes. So groups of ribosomes in the cytoplasm that are making protein. As you can see, we've got lots and lots and lots of ribosomes.
So why would the ribosomes be grouped? Why would you get polyribosomes?
Because where is the code that tells the ribosome what proteins they have to make? Another question. Where is that code?
Or what is that
What
code?
sort of molecule is that code? Yeah.
An answer from Ben.
Yeah.
So Ben says it's mRNA, and Rose says mRNA as well.
Very good. It's mRNA. It's a long, thin molecule. Yeah. It's a polymer. So it's a string of beads kind of thing. Yeah. So the ribosomes attach to one part of the mRNA, and they go on reading the message. Yeah. And once one ribosome attached to the beginning of it and started moving along and reading the message, then the next ribosome attached to the same molecule and then starts moving along. And then the next ribosome attached to the same molecule and starts moving along. So then you've got, imagine like a cable that lots of people are holding onto this cable and kind of moving like this. Yeah. So that's why you have the polyribosomes, because they are attaching to a long messenger RNA. Yeah. Oh, this is a nice mitochondria. You don't always catch them in the right position. Nice. Does anybody have any questions? I think I'm getting to the end of it really, and
Yeah, the last 10 minutes or so now. And if you've got any questions or things you'd like to see,
or things you didn't understand, you'd like me to explain again. And you'll see that.
Yeah.
Oh, beautiful, crispy, all stacked up together.
just take it of your mind.
No questions?
Not really, no.
Not really.
Harmony says, how are the proteins made from ribosomes loose in the cytoplasm, processed and folded?
Oh, so there are all over the cell, yeah, there are some proteins that help other proteins fold. They are called chaperones. So we've got chaperones all over the place in the ER, and we've got chaperones in the cytoplasm. They help proteins get to its proper folded state. Yeah.
So it's not not just in the endoplasmic reticulum, they're
No,
also everywhere
yeah,
else.
we've got chaperones everywhere as well. Proteins fold. And although the actual information for the protein folding is in the protein sequence itself, a lot of the times the proteins wouldn't achieve the proper folding, folded state if they didn't have these other proteins help them. Basically. Any more questions? So you can see how tight the membranes of these two cells are, kind of how close together they are, because this tissue is lining the inside of the gut. And so the cells have to be really close together so they don't let anything that's in the lumen of the gut go inside the person without going through these cells. So they kind of filter out what goes inside the person.
Ben's asked us a question of how does an electron microscope form the vacuum?
Ah, very good question. So the electron microscope has a pump, yeah, that basically actively pumps atoms out of the column of the microscope. And this pump is right at the back there. It's a little bit noisy. You can't hear the noise because you're a bit far away, but the pump's a little bit noisy. So all the time that we use the microscope, there's got to be this pump. The other thing that we have on transmission electron microscopes is something called, it's a liquid nitrogen device called an anti-contamination device. So this also improves vacuum. So right up here, we have a tank that we have to fill up with liquid nitrogen. Every day when we use the microscope, we have to fill up this tank with liquid nitrogen. The liquid nitrogen then cools down a plate inside the microscope. And this plate, very, very cold plate inside the column of the microscope, serves as a trap for molecules. So this molecular trap also helps to improve the vacuum of the microscope. But what's actually doing the job of really creating the vacuum is a pump that actively pumps out. It's a vacuum pump. It's called a vacuum pump. It pumps out molecules from the column of the microscope.
And a thing that's interesting is that if you were to, when you install this microscope, there obviously wouldn't be vacuum inside. And because the vacuum needs to be so perfect, it can take almost a week for it to reach perfect vacuum from starting.
Yeah. So it's because electron microscopes have the thing that generates the electron beam is called a filament. Yeah. The filaments, there are different types of filaments. This electron microscope has a special type of filament. And this filament is a single crystal of something called lanthanum hexaboride, lab 6. So this single crystal, the tip of this crystal is very, very, very small. And if this crystal is not all the time in vacuum, the tip of it can get eroded, so then it stops generating a nice strong beam that we use for the microscope. This particular type of filament, it needs very strong vacuum. So some filaments don't need such strong vacuum. This filament needs quite strong vacuum. So here we've got another nucleus with a nice structure in the middle here that we call the nucleolus. Nucleolus generates a lot of RNA to the cell.
And what are those black bits in the nucleus?
Yeah, so these are a little bit of condensed chromatin, so we call it heterochromatin.
What's chromatin?
So chromatin is when the chromosomes are all wrapped around proteins, they form this structure called chromatin. Oh, okay, so I think we might wrap it up.
Yes.
Go back to that nice little bit of VR that we found right at the start. So I hope everybody enjoyed it. I hope you understood more or less how this microscope works and how we look at cells, at the inside of cells using electron microscopy. And thank you so much for coming. And thank you so much for the participation. I was very impressed with everybody's answers. I can tell that there's some really enthusiastic cell biologists in the audience and, well, budding cell biologists in the audience. So Thank you so much for coming.
Well, people saying thank you to you as well.
Thank you.